So this is an interesting lab because it's very intense what they're wanting us to do, and it would definitely take a lot of time, which is something that's kind of at a premium for us. So we're going to change what we're doing a little bit. This is the cell division, mitosis, and meiosis lab. It's investigation 7. It starts on page 83 of your lab manual. And so what I've done is posted on my website a very condensed version of what this lab wants you to do. And we're going to do a lab where we actually look at cell communication and how cell molecules are going to help to signal for the cell to go into this cycle. So what this lab is wanting you to do is actually look at a couple different resources. It is wanting you to do some background reading, which I agree with you doing. So if you look at the investigation 7, I ask you to read pages 83 to 85 and summarize. And what we mean by that is that really, this is a really good review. And so you do want to take some time to read and summarize. It's okay if it doesn't get done tonight. That's okay. But at some point, this is a really good resource to look at. Talks a little bit more about CDKs, talks about those checkpoints. It's a really good review. Once you get onto page 86, there are 8 questions here. So one of the things that I am asking you to do is read through these questions, pick 3, and then answer them. So of course you want to pick the ones that don't come the easiest to you. Ask me for help. Ask your group for help. Hopefully you pick ones without asking your group so that maybe you all have different questions to talk about. But the key is that you're able to actually really think and process it. One thing I would say is I don't know that you should really be expected to figure out exactly how many divisions, but if you're super good at math, then go for it. If you look back at the sheet I'm giving you, I am asking you to look at some actual cells. So one of the things that the lab asks you to do is it asks you to actually create your own slide. It wants you to take real onion tips. It wants you to do this many series of chemical reactions to get to the point where you can see the chromosomes in an onion root tip. I'm just not sure that's an effective use of our time. So instead of doing that, I'm providing you with these pictures. I do want you to try to label this, like when you look at this, what do you see? What does this diagram represent? And the lab manual gives you a little bit of background, and we can talk about it tomorrow as well. Labeling this and any stages visible that you see. It does give you a hint if the cell appears blank, it's because the DNA is relaxed. So explain what that implies. I'm sorry, it should be implies. And what the kind of implications for the bigger picture are as well. And again, we can talk about this a little bit more in class. The next part of the lab, it does actually really go into a cool experiment on the effects of mitosis, what might increase or decrease the rate of mitosis. But again, this is a really long lab, and I'm just not sure that the amount of time that it takes is useful. The next part actually looks at cancer, which is something that we would definitely like to talk about a little bit more. However, it goes into some aspects that I just don't think are really relevant to you. So we're going to talk about cancer and how you can kind of apply that a little bit later. So skip that part for now as well. Part 4 is modeling meiosis. It wants me to give you clay and how do you create the chromosomes and do crossing over, and that's what you're doing when you're doing the actual drawing, which is something that you can take home with you versus a model of clay that is not going to go home with you, so you can't study. So I think that the drawing is much more effective. The last part is actually really important. This is looking at a fungi, and it looks at how the fungi makes spores. So here we see a little alternation of generations, diploid, and then we see haploid, and so it is looking at how this fungi creates spores. It turns out that when it creates spores, we can actually see crossing over. And they're not showing you a picture because they want you to actually see it. However, again, this takes a really long is giving you a little bit of background. You've got two diagrams here. One of them shows crossing over, one of them does not. I'm asking you to label which one is crossing over and which one is not crossing over, and then use other vocabulary to label the diagram. And then this is actually a picture of what you would see under the microscope. And so these are the spores. It shows you what crossing over looks like, what crossing over does not look like, and it asks you to tally it up. So we're going to use this. If you can print in color, then this will be a lot easier for you to see. If you can't, if you'll just let me know, I'll try and have some color ones printed up. So this is a very quick summary of what we're going to be doing. The bigger thing that I'm asking you to actually do is read through this lab manual on cell communication. Da, 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 da. And it's fairly long, and so we're probably going to need to go over it a little bit in class. One of the big things that I'm asking you to look at is if you can read the background and really look at this diagram. I don't have a digital copy of this. Let's see if I can make it come up so you can see it. This is the diagram that I really want you to see and try to look at and understand. It's blurry on here, but you have your own paper copy. So reading through this to try and get a better idea of the actual lab that we're going to do with two different types of yeast and the way that they communicate with each other, that'll be the actual lab that we'll do. So thanks for your time tonight.